All right, so we're starting with 4.1, and what we are looking at here, that just know that what are the different types, that you do have two different types of interaction, either cells or the cell with the matrix. So know that they are visible by the fluorescence microscopy, and this is one of the most important slides, so that you have an overview of that. Just make sure that you have a handy copy of this, so that this gives you a kind of an overview of the idea that what is what, and that this is a good slide to keep handy. So after that, this tells you all the different ones, the junctions, and we'll just individually tell that what is important. Definitely, we'll talk about that. So again, tight junctions, know about that. And then I just want to bring one of the things that even up front, you can see right here, I just made a separate screenshot of that just make sure this is the anchoring junction summary. And there will be some of these questions which are, I know that it is like a just memorization question, but it can give you some of the points. So if you just keep this chart handy, and it is just that what they are made up of. So all you have to do is just look up quickly, even if you don't remember them individually. So this is a slide or a printout that you can keep handy, give you all the different ones. So that will be helpful. So let's go here, what we have. So one of the type of question that can be asked, say from here, you have a fluorescent tracer molecule, and if you have a gap junctions, you can be given a couple of different conditions, that you inject that fluorescent tracer here, and in one, so let me do one thing. Okay. So you have use the fluorescent tracer molecule and allowed that to diffuse, and then you are given condition that what is going to happen. So suppose here you have a tight junctions. You can be given that there is nothing like this, or the fluorescent molecule has passed in here. So that's basically what it is. Or something, it could be that everything has passed here, there is nothing in there. So remember that if you have a tight junctions, nothing is going to pass through them. So that's a type of typical question that can be asked. All right, so let's see. So this gives you an idea that through tight junctions, nothing will pass. Gives you an overview of that particular area. These are some of the previews from that, what it is. And again, remember that this just emphasize the function of the occluding junction is to maintain the gradient so that all these phenomena happen properly. You do not individually need to remember the mechanism of any of the transport, because that we have studied beforehand. So that's the important part. All right. So again, definitely remember these JAMP, Claudine 1. Remember that sometimes question could be asked is just Claudine. Claudine and Claudine 1 is the same thing. So don't think that instead of Claudine 1, if I just have one of the answer choices, Claudine, it is not a trick questions. Claudine is a family. There are many different types. That is the same thing. And occluding is also one of those. These questions could be just remembering that what they are, that type of questions. You will see these questions. All right, so definitely you will remember this from the chart, so that's not a problem, where you find the intermediate filaments in the desmosomes, whereas a cell-to-cell and cell matrix, you find mainly the actin. And this is an important concept, so remember that you have a transmembrane linker protein. Then you have the plaque proteins. These intracellular attachment proteins are also called the plaque proteins, which will attach to the cytoskeletal filament. Could be either actin or intermediate filament. So just an overview of that idea. So here you have an adherence junction, gives you an overview of that how this adherence junctions function. I think that this is a very broad belt, so remember that. And the proteins involved. Also, you have this myosin 2 filament. You find it in the adhesion belt other than the actin. So this is one of the important part that is there. So just remember that this particular example, if I just bring here, so this is a tricky question. If you see right here, the cell-to-cell anchoring junction adhesion belt, actin, transmembrane linker protein, cadherin, catenin, vinculins are the attachment protein or the plaque proteins, and ligand, which is the other cadherin in the neighboring cell that is attaching. But see that here, your myosin 2 is missing. So this could be an information that you have from their slide, that adhesion 2 also do have, other than the cadherin, catenin, or vinculin, which are in that chart, you also do have myosin 2. So that you can see. All right, so this is also another idea that how the formation of the neural tube happens, basically. And just remember that you have this adhesion belt, which squeeze the cell, top of the cell, into kind of a being tapered. So the top of the cell just becomes a little bit tapered, and that makes that neural tube. It's very straightforward. So remember that what are these focal contacts, and remember the proteins that are associated with the focal contacts. So again, that's very important as well. So the intracellular attachment proteins are talin, alpha-actinin, and the transmembrane protein is integrin. But again, this talin and alpha-actinin attach these to the actin. So this concept. So again, as I'm repeating, integrins are the transmembrane linker protein. These are the proteins which are outside, attached to the matrix. Then alpha and talin alpha-actinin are the ones which are inside, and which is not shown are the actin to which those intracellular attachment protein bind these focal contacts. And remember that lamellipodium is right here, is the leading edge of the cell where it is moving. And that part, so here the focal contacts are being made on this part, whereas the focal contacts are broken at the trailing edge. So these are some of the things. So this is a same thing from the previous chapter. Desmosomes a little bit. So again, remember that the intracellular attachment proteins are often called as the plaque proteins or cytoplasmic plaques. All of these are the same name. So these are the attachment protein. Know these. All right. Autoimmune disorder, but just know the name and how it happens, and mainly affect the skin desmosomes. So main idea is that due to what this happens. So autoantibodies against the desmoglin, which is the one of the proteins which is responsible for the attachment protein. So remember that transmembrane linker protein. So that's desmoglin right here. So think about that if you have this transmembrane linker protein, which is desmoglin right here, and if you have an antibody against that, so what is happening as the cells are dividing, these cells are being made, and if the antibodies are binding to that, the other side's cell will not be able to attach to them, and there will be no cell-to-cell adhesion. So basically, the structure will be very flaky. These cells are going to fall apart, and that's what we see in the in this autoimmune disorder. All right, I think these are some of the, so remember that your keratin is an intermediate filament. That's also a very important thing that we have learned. And here you can see that the hemidesmosomes and the desmosomes different. Desmosomes are in between the cells, where the hemidesmosome attach the cells to the basal lamina or the basement membrane. So that's the difference. This is the important one that I just made that definitely keep this handy. That will help. All right, gap junctions. So let's see here. All right, so gap junctions is a communication molecule. So here you can have some interesting question. So suppose I just give you a picture of like a 3 cell, 1 here, 2, 3. So let me just draw. So I just give you a picture of 1 cell here, and then I give you another cell here, I give you another cell here. Now what I do that says this is your number 1, this is your number 2, this is your number 3 cell. I inject a dye right here to this cell, which I'm telling that it is a green fluorescence protein dye, which is the fluorescent tracer molecule. So if I inject that dye to this cell, then I give you the choice that suppose this is a fluorescent green colored dye that you are seeing, and then I just give you the choice that it will be only in this cell, it will be this cell and this cell, it will be this cell and this cell, it will be all of them. So what do you guys think that if I just inject a fluorescent tracer molecule like a GFP protein tag and look at them under the microscope, what will happen? Anyone?
It will only be in the center cell, cell number 2.
Yeah, that is correct. It will be only in the center cell, because think about that the only molecule which are passing 1,000, maybe 1,200. So you will see that you probably have a question in your practice quiz that it just had a question about 1.2 kDa. Yeah, so maybe 1.2, 1.5. But again, if it is 5,000 or higher kilodalton, it will not going anywhere. So the fluorescent dye will remain in here. Now, just to confuse you, I'm just saying just to confuse you, I can tell that I am injecting a calcium molecule, a calcium, and along with that, the fluorescent dye, along with that. So if you see that inorganic ions, amino acid nucleotides, so your calcium is an inorganic ion. And I tell that I'm just injecting a fluorescent dye along with that, which is the GFP and calcium. And in that case, what will happen? Will you see any difference from what you answered previously, you guys think? No? Someone?
No? No difference?
No difference. Yeah, that is correct. There will be no difference, because think about that. Your calcium, sodium, or any of these inorganic ions, they are not fluorescent. I mean, like when you're looking at the the green fluorescence, you will just only see the green fluorescence. Those are a calcium molecule by itself is not going to fluoresce. Now, remember that sometimes previous things do not line up, and sometimes you may think, because we talked about that there is a dye which can sense the calcium concentration when we were talking about this tracing the calcium. So that is an entirely different thing. It is a dye which senses the calcium concentration and fluoresce, but calcium by itself or sodium or any of the inorganic ion or amino acid, they are not fluorescent. So just to confuse you, I can tell that I just have added an inorganic ion amino acid nucleotides, which are well below, say, 1,000 Dalton or 1 kilodalton. And along with that, I just added this fluorescent dye. Your answer will be the same, because you only are concerned about the fluorescent dye, which will fluoresce, not the other molecule. Yep, you're going to see this question. I already told you one question straight. All right, just that overview of that what are the proteins? These are called the connexin. One of the interesting part that think about this, that the pores need to line up for these movements. So that's one of the important things. I think that other than that, there is nothing. So here is also another thing. So think about that's why I said the calcium along with the fluorescent. So see that high calcium right here will shut this channel right here. On the other hand, a low calcium or the high pH will open this channel as well. So on the other hand, even if you think that I am adding calcium, and again, this is usually what happens, that even if you high condition of the calcium, not only the GFP will not move, but if you have a high calcium, the gap junctions will be closed. So you have a kind of a double whammy in there, that even in condition of high calcium, it will be closed, so it's not going to open. And along with that, we have talked about a very large molecule that will not pass. So even if you can correlate the calcium with this, the answer should be the same. It does not violate this law. All right, so let's go to the next one, I guess. So let's start with all right, so So 4.2. All right, so very basic idea. Just know the homophilic, heterophilic binding, what they are, and always remember that the ECM is heterophilic. So this is an important slide. So definitely remember that the cadherins, the different types of cadherins, and the IG family and CAM, and these are homophilic, and what are the heterophilic? So important names. So this is also another slide that you can keep handy, just to make sure that you remember those. All right, so here you have all the different types of cadherins. And again, I'm not going to ask that where they are found. Like that would be kind of too much. But one of the important thing that your EP and N cadherin do not interact with each other. So yeah, so I'll come back to that. So here, this is the idea that this could be a question that you can see that only the E cadherin, P cadherin, and N cadherin interact with each other. So EPN will not bind. So this is also a very important question. You can have an application-based question. We can just directly tell that we have these different cells, just how the experiment has been done. Some cell and these three different types of cells mixed with EP and N cadherin. And if you're just seeing them, whether you will see any kind of interaction with the other molecules or something like that. You may see that E, P, and N is labeled with a particular fluorophore to check those, like an antibody, which could be, say, for if you see that there is E cadherin, there is a red color. For the P cadherin, there could be a green color, or say N cadherin, there will be a yellow color. And when you have those conditions, you may have to predict that you are seeing under the microscope in between the cells only the similar color. So if I just say that in between the cell, you are seeing red color, that is correct, or a green color, that is correct, or a yellow color, that is correct. But if I see that I see yellow and green, no two different colors, if the cells are being attached to it together, will not happen. So if I say I'm seeing yellow and red, or yellow or green, that will be untrue. So that kind of a little bit of application-based question can be asked. All right, so here you know that the structure overview of that, 3-4 calcium binding domains, about 100 amino acid each. And then this is from the previous one, but definitely remember that what are the plaque proteins? These are the catenin and the vinculins, which attach the cadherins to attach the cadherins to the actin. So one of the important thing that remember right here, there is a lot of proteins right here. So this is also another slide or picture that you can keep handy that will also help. So you can see that for the cadherin, you have the similar homophilic interactions against each other, and how it is formed, very straightforward. Lots of different types of proteins. So those are important. And again, just names, really, not anything mechanistic. I don't think that that's it. But this is also another thing that you can keep handy. We already talked about that. So think about that. That this is just a modification of this experiment, just given to you in a little bit different form, where you see the colors in between them, and you may have to predict that type of phenomenon. That's it. All right, calcium is very important. Just only remember that it is not only for the Ig superfamily. Calcium is a very important molecule for all of them. So just remember that that is a very important phenomena. Even for the cadherins, calcium is very important. Now we come to one of the question that you can easily remember, that we talked about that when we try to take out cells that is growing in the culture, they attach to the surface of the culture vessel, which is your flask or petri dish. These are all made up of plastic by these attachment proteins, IGs, the integrins, and all these, they're all attached. And there are lots of these calcium-dependent attachment. So what you do, that you use the EDTA, and remember that the EDTA is a chelating agent. So what it's going to do, that there is enough amount of calcium in the media, in the cell culture media. So the EDTA will just capture them, and they will not be available for any of these which are dependent, many of these are dependent on the calcium. So they will disrupt these camps or the cellular adhesion molecules, and that's how the cells will come off and you can passage them. So this is a kind of like a setup question that can be asked with that. And also along with that, remember that we will come to that. Let me see, the protease question. We'll come to that in a second. So again, remember that the integrins here is also calcium, because when we talked about, say, the calcium for Ig superfamily or for the cadherins, you have the cellular interaction. But when you have these focal contacts that is being, or the hemidesmosomes, those are sitting on the surface of the cell, and they are attached to the matrix of the plastic by the integrins, not the cadherins as such. So what happens there? So you can easily see that all of these CAM proteins need the calcium, so the EDTA will have the same function. So this is your structure of your integrin. Again, you do not have to memorize this whole thing, but just remember right here, I will just only point out to the right here, this not only the T lymphocyte, but all these leukocytes will have an alpha L beta 2. So I can just tell that, say for an example, again, this will not be a memorization question. I say that, say for an example, that I mutated this beta 2, mutated this beta 2 protein, so it cannot interact with the alpha chain. And I will just come to that what it means. So right here, you can see that that is the beta 2 chain, say for example, for the leukocyte, and this is your alpha chain. So remember that for the matrix binding, you need both the partners. And that will be right here. You can see that right here is the structure of the integrin. And you have this both alpha and the beta, and both is needed. So if I just give you an example that where you have this beta subunit mutated in such a way that it cannot interact with alpha, so it's non-functional, remember that then this integrin molecule will not be activated. And we'll ask that what kind of thing can happen to the leukocyte in the extravasation. And we will come to that in a second. So the next thing that you can just see that how regulation happens. One is the intracellular signaling cascade by different proteins. That's one of the things that can happen. Conformational change of the receptors through the receptors, or sometimes the phosphorylation itself, like the serine residue of the beta chain of the integrin can will make so just a phosphorylation of the beta chain will keep or disintegrate the whole complex as such. So these are some of the regulations. All right, so structure is not needed. This is just for illustration purpose. So you can see that this is a kind of an interesting thing that in inactive integrin, they are kind of like like this. They are bent down, whereas when they're activated, they can just become a little bit straightened. So this is a typical structure of the activation that is being seen in a cartoon form. So this kind of flips up this portion. So that's the activation. So here is, you can easily see that also another thing that, again, remember that I'm not going to ask any of these signaling questions. So you do not have to memorize any of these PI3K, GERB2, which happens after that. Just the take home message here that if integrin can serve as a signaling molecule, so if a ligand bind, it can just cause ultimately the cell migration gene manipulation, but just transcriptional activation. So just overview of that. I'm not going to ask any detail of this. All right. All right, good. Very nice. Any other thing? Okay. All right, blood cell extravasation. All right, so here is an interesting thing. So just make sure that you see this and understand this properly, because you are going to expect a couple of questions from here. And also there are some of the videos that is posted below the lecture video. So take a look at that and you will be able to see one of the real microscopic process by which a live cell is being shown, that how the extravasation is happening in there. So just to remember that it is step-by-step process. So one of the thing is very important, and this also came up when I was discussing this with another student who had, he had a question, that when you have this inflammatory response from the injured and infected tissue cells, these are the different types of interleukins that we're not going to detail. So when they act on these, the first thing what's going to happen is the vesicles that will have the p-selectin. It's already synthesized and inside the cell, but this signal will traffic them on the cell surface, and the first binding of these selectin ligand, which are the mucin-like CAMs, with the selectin, this is a pretty low affinity. So it's not tightly bound. So the function for this is that leukocyte earlier, when there is no signal, the leukocyte will just pass quickly. And I highly suggest you to watch the video, because that clearly shows that when there is no activation, the leukocytes are passing through the blood flow very fast, and then you will see how it rolls. So the rolling is clearly shown in that real microscopic picture. While it is doing that, it will find a platelet activating factor that will bind to its receptor, and now it will signal the integrins to be active. And now these integrins will bind to ICAM1 or ICAM2. Both of them are the receptors, and this is a heavy or high affinity binding. So again, remember, selectin and selectin ligand mucin, that's a low affinity, whereas the integrin is a high affinity binding. It's going to pull the leukocytes very close to the cell surface, and when it finds a space in between the cell, it will squeeze itself out into the subendothelial tissue or the connective tissue space. So the question that was asked here, that you can see that there will be cell adhesion molecules in between these cells. So how come the leukocyte can squeeze itself through here? So there will be two kind of understanding. This is not required for, I will not ask this question mechanistically, but just for an understanding. So remember that your intestinal cells, which are the same type, you have these epithelial cells in the intestine, they do have these tight junctions and the adherence junctions in between them. So they are really, really tight, and it's almost impossible for any cell to pass through those junctions. It's almost impossible, because those cells do have the polarity to maintain think about that, suppose this is the apical surface and this is the basal surface. To maintain the polarity, you have those tight junctions, and below that you have the adherence belt, and no cell can do that. But here what you have, the endothelial lining is just one layer of cell, one layer. That's the important part. One layer of these cells, and in between them, they do have cell adhesion molecules like adherins, but there is really no tight junctions or the adherence belt. And why it is so? Because of these phenomena. And then on top of that, what can happen when you receive this inflammatory response right here, it will, by the mechanism of this interleukins, it will make a change in the cell so that the cell adhesion becomes less. So there will be much less cell adhesion. Think about that. The cell may shrink a little bit, and it will make some space in between the cell so that the leukocyte can pass through them. So that's basically one of the response of the endothelial cell for the passage of the leukocyte. And that does something else as well. Think about this, that as simple as that, if you just have a mosquito bite, you just get a bump in there. So why there is a bump? So the reason for that, it is the same thing. If you just have a mosquito bite, it will cause an inflammatory response. And then this is the same thing that's going to happen to the capillary of the bite site. And along with some of these leukocyte, which is exactly not needed that much in a mosquito bite. But the other thing, what happens when the cell shrinks right here, there will be leakage of the blood plasma. So the watery part or the fluid will just come in here. That will be in the subendothelial tissue. That gives you the bump. So you can now tell that why an injured tissue or infected tissue gets inflamed or just get swollen due to excess amount of fluid because of this, because these endothelial cells allow the water part of the plasma to leak into the tissue space along with the leukocyte. All right, any questions on this? Anyone? Good. Okay. So that's, I think, that overview of that. That will do it. So let's see what we have next. All right. So let's see what type of. All right. So just know the very basis of cancer and what are the phenotypes of a transformed cell. Remember that this is very, very important. When we just are talking about a tumor cell, transformed cell, neoplastic cell, they all mean the same. So again, remember that what are the different things that is happening into the cell, and we'll ask some questions in there regarding those. Let's see. So this is also another idea that in a benign tumor, usually there will be a fibrous connective tissue which is covering the cell. And here there is a very important concept. Remember that even in the benign cells, when a tumor is growing, that is still, they are neoplastic cells. So that means that there is some problem going in there, but they are still contained with the fibrous connective tissue and not going out of control. And sometimes it can become bulky or something, but the thing is that just don't think that the benign tumor are not neoplastic cells. There is some problem of the cell cycle control by some oncogenes or tumor suppressor gene in there. But what happens right here, that these neoplastic cell in the malignant or the metastatic tumor do have some of the extra type of genes, and we already talked about that there are 3 to 7, at least minimal 3, maximally 7 or more needed to be mutated, and they mutate in such a way that that anomaly or the aneuploidy of the chromosomes will cause these fibrous connective tissue to break up, and they can just move in different directions as well. And what is metastasis? Definitely know that. So these are some of the difference that, again, so a good question is the comparison of your transformed neoplastic cell with this. So that could be definitely a question. So remember that you not only require the medium, but you also, the normal cell is anchorage dependent and display contact inhibition, whereas none of these are required for the transformed cells, so they can pile upon each other. So you will definitely find these questions on the that what are the different types of behavior that the transformed cells do exhibit, definite questions. So this is also another thing that here you have an in-between lines in there. And I highly suggest that definitely underline this and remember that. What are the adherence abnormalities? There will be some questions which are kind of a difficult in-between line. It's not difficult, but the thing is that it is easy to miss. And one is that reduced external coat of fibronectin. And where is this fibronectin? That I will just explain that. See, right here, you have the fibrous connective tissue. The fibronectin is right here. And how you correlate, if you remember and go back to your previous chapter where we talked about the anchorage dependence, and this is sometimes becomes a little bit difficult for some. Let me just go back right here to correlate two different things. So right here, if you just see that this is, we are looking at the slide 4.1 part 2. A heterophilic interaction in between the integrins and fibronectin. This fibronectin is an ECM protein. So that's an ECM or extracellular matrix protein that you have learned here. And you are getting the same information right here that you have this extracellular matrix, which is a fibrous connective tissue. And in here, you have this fibronectin. So you can see that you can correlate three different things from three different slides, and sometimes those type of questions. So remember, understand the connection so that it will make the questions answering easier. Another important thing that we talked about while doing the lecture itself, see that the plasminogen activator cause increased extracellular proteolysis. That means that it will cause these cells to secrete some of these proteases. Again, I'm repeating, the plasminogen activator will activate some of the proteases. What it will do, that it will digest the extracellular matrix. And once these extracellular matrix proteins, some of them are digested, they will not be bound to them and they can come off the surface. And the similar process that we did in the cell culture, when we culture the cells in the mass, like regular cells. So for the anchorage dependent cells, like right here, suppose we are culturing the cells and now they are confluent, means that they are all touching each other and there are contact inhibition, and I need to passage these cells. I need to split this up and put it in a new flask. At that time, we add the EDTA and trypsin solution to that so that these cells will come off. So again, we talked about the calcium. So think about that. The calcium will disrupt all the cadherins, integrins, and IgCAMs. So the interaction in between the matrix as well as the in between the cells will be broken. The next thing is also the protease. So what happens that there are some proteins secreted by these cells which are attaching themselves onto this surface of these matrix. So these are produced by these proteins. Say for an example, what I'm saying, that right here, suppose these are the fibroblasts, and this fibronectin, collagen, we did not talk about many of them, but all these different types of extracellular matrix protein, they are going to be secreted by these cells and they will attach themselves onto the plastic surface to which these proteins are attached. Now, when you add the trypsin, it will disrupt those proteins or digest those extracellular matrix protein, so they will come off. On the other hand, for the cancer cells, the cells produce this plasminogen activator, which will make these cells to produce some of the proteolysis to do the same that you were using in that. So just make sure that you understand that concept. All right, so these are some of the growth division abnormalities. So remember that what is the focus of the cell, that is important, and they are immortal and then can cause tumor when injected to susceptible animal. And what we mean by a susceptible animal, remember that a susceptible animal means that they are immunocompromised, because even if there is a tumor causing cells, if you just inject it to the animal will treat that as a foreign cell and its immune system is going to kill it most of the time. There could be some unfortunate situation, but most of the time. So that's why you need an immunocompromised animal model so that the tumors can develop. And this gives you an overview of that. We talked about that. There are different types of mutations, like a nude mouse where they cannot produce any T cells. This is a skid mouse where this is not needed. I'm not going to ask that, but the skid mouse will not produce any T cells or the B cells. So suppose here you can ask a question that I take a skid mouse and I take, which is like this, no T cell and B cell. I take a normal mouse and then I inject these transformed cells to those. And then I just give you a chart, the chances of getting a tumor in the skid mice and the normal mouse. Remember that the skid mouse percentage of tumor will be always more than the normal mouse. So if I have that thing like this, is that the normal mouse, 70% of the normal mouse produced a tumor, whereas 30% of or 25% of the skid mouse produced a tumor. That will be absolutely wrong. But if there is a choice that you see that 80% of the skid mice produce the tumor, whereas less than 5% produced that in the normal mouse, that will be the answer of choice. So that's something like this. Or if I just say that the chance, so 50% each of the skid mice and the normal mouse produce tumor, so that will be also a wrong choice. A normal mouse, chances of getting a tumor in a normal mouse is much less than a skid mouse. So just make sure that chances of getting a tumor percentage-wise will be higher in a immunocompromised mouse like a SCID mouse. And I will describe that. Suppose you have a SCID mouse, it cannot produce T cells and the B cells, and it is immunocompromised. And you have a normal mouse, which has all the T cells and the B cells and the normal function, and I will describe the experiment and tell you that the chances of getting a tumor, which is a kind of a correct thing that you choose. So that kind of question, you can see that. Then we are going to talk about the proto-oncogenes and genes. So here there is an interesting thing. So suppose we can ask some of the, again, you do not have to memorize any number, but you can see a curve right here. So say for an example, here you have overview of per 100,000 persons and an age group. And I just draw a curve like this and then tell you just similar type of curve. And then tell you that, okay, all right, this is the percentage of, say, colorectal cancer in women. So here you have this patient numbers per, say, 100,000 occurrence. And here you have an age group in this. And I say that this is the percentage of colorectal cancer in the female. Or say this is the percentage of esophageal cancer in men. And which of the following will you infer from that chart? So the take-home message is the same for all of these. Remember that what you are seeing, that when no matter what, whether it's a men, women, or whatever type of cancer it is, the take-home message is that when they were young, the cells have not divided that much. Chances of mutations are less per gene, and that's why you do not see them. As we age, more and more mutations occur, like this is the number, more and more mutations occur, and that is the hypothesis that is proved by this type of thing, that as we age, more and more genes get mutated, which leads to the cancer. So just try to understand that what will be the outcome of the curve. Like if I just say that it means that the chances of colorectal cancer is more in women than men, that does not make sense. Or say that if we just say that women only get colorectal cancer after the age of 70, that's not right. Or if I just say that that means that the youngs have a different food habit, that's why they do not get the colorectal cancer when they're young, when due to the food habit change, as you age, the colorectal cancer is increasing. That's not correct, but this is the whole thing, that you have accumulation of mutation that can lead to colorectal cancer as we age. More than one mutation is needed, which is shown right here. So again, that this just gives you that individual occurrence versus synergistic effect. When you have both of them mutated, what it can cause. All right, then we're going to talk about these quickly, the other two parts. Let's see. All right, very, very important slide. So definitely remember that what are the different ways that an oncogene, but with a gain of function, can lead to a proto-oncogene, which can potentially cause the neoplastic phenotype or cancer cells. So all of them are important, and know that what it is. So just make sure that you understand that sometimes the words are used. Suppose here we are using the word like a deletion point mutation in the coding sequence. Here is one interesting question that can be asked, and probably it will be asked. So suppose we are just giving you an idea that we have a mutation which is a kind of a nonsense mutation. That means that somehow due to a deletion or a mutation, the coding region of that protein is completely changed, so that made a mRNA and a protein from that mRNA is completely different and is completely inactive. Now remember that if the protein is inactive, that is being produced due to the mutation, it cannot be a proto-oncogene. Remember that. This is a very important concept, and you can easily get carried away because I said that there is a mutation which produces a defective protein. And a defective protein which has lost the function will not act as a proto-oncogene. So make sure that you understand that concept. It has to be hyperactive or constitutively active. It has to be more active, not inactive. That's the important concept for your oncogenes. And this can happen due to the same protein, which is not hyperactive, but it just produced more by this gene duplication or amplification. Or it is translocated, that gene, under a very strong promoter, which is constantly on, not regulated. That can cause that. Or it can fuse with some other proteins, which will cause the signaling from this particular protein constantly, constitutively hyperactive fusion protein. So these are some of the things. And you can easily see that the class 2 is the receptor tyrosine kinase. Here is an example that how it can constitutively become active. It doesn't even go into the plasma membrane. This fusion protein, this weird fusion protein, remains in the cytosol and constitutively active and implicated in some of the cancer. So this whole pathway will go on, and ultimately it will produce the proteins which will drive the cell cycle, and that's no control of the cell cycles basically is the neoplastic phenotype. So here is also an EGF receptor. So remember that all these EGF receptors are the family of receptor tyrosine kinase. They work the same way, it's just different way. So this HER2 receptor is implicated in breast cancer, where a valine is changed by a glutamine. This is a very specific question, but it is very bold and important in here. So remember this one, a little bit of memorization question. That you have a HER2 receptor, which is implicated in the breast cancer, where a valine is converted into a glutamine, which allows the autodimerization of this protein without a ligand and cause constitutive signaling. And here you see that a deletion of this caused this. All right. RAS, very important. Remember that. The only take-home message that no matter what kind of mutation happens, if GTP is bound to RAS, it is constantly on. If it cannot hydrolyze the GTP by the help of GAP or just by itself by any mutation, you cannot turn it off, means the RAS is constantly signaling, and that can lead to transcription activation of many of these cell cycle proteins, which can drive the cell cycle uncontrollably. So that's basically the pathway. So if this is on, all RAF, MEK, MAP kinase to transcription factor and growth will be on. And the only way that it is on, if it cannot hydrolyze the GTP. Right, let's see. Very interesting idea. Remember that. All you have to remember that these are the portion of the antibodies under a constitutive promoter. This is an immunoglobulin gene promoter, enhancer or promoter. So this is constitutively active, all the time signaling. Due to the translocation, MYC sits just after this. And now MYC, which is highly regulated, MYC is only expressed in certain stages of the cell cycle to drive the cell cycle. Other times the MYC is off. So MYC is a transcription factor itself, a transcription factor for the cell cycle. Most of the time off, only on in certain time. Once the translocation happened, this immunoglobulin gene enhancer is constantly on, and that will drive the MYC constantly on, and that will be uncontrollable cell growth. All right, let's go to the last one right here. So RB, first gene, retinal tumor. And when we are talking about the tumor suppressor gene, remember that for the oncogenes, so the proto-oncogenes, it is a gain of function. And here we need a loss of function. And for the loss of function, it is recessive. So 2 copies are present in each allele of our pair of chromosome, and both needs to be defective for the tumor to appear or neoplastic phenotype to appear. So this is just an idea. So normal cell, you have a normal allele, mutant allele defective. So remember that just before the cell division, these chromosomes needs to be duplicated. So this will duplicate in this will duplicate in this. And then what will happen that if you have 2 cells, equal number should go. One should go to one cell, another should go to another cell, which happened here. Each cell received one of this, and one of this should go to this cell and another cell. Unfortunately, due to the mitotic spindle aberration and motor protein aberration, one cell received 3 of them, and although this died, this survived, and that can lead to an unfortunate event where both of the loss or the lot of heterozygosity. Remember that this cell is heterozygous. So heterozygous means that this normal allele is still producing the protein which can control the cell cycle. And here you have a complete loss. It cannot control the cell cycle, will cause the neoplastic phenotype. And that can happen due to the mitotic recombination as well. Recombination within the homologous chromosomes are common. So remember that what we mean by the homologous chromosome. So suppose this is one chromosome and it's going to be one pair. So remember that your chromosome number 1 is 2 in number. Chromosome number 2 is 2 in number. So you have 23 chromosomes, which are, well, 22 chromosomes, which are in pair. And then you have either XX or XY, depending male or female. So for each chromosome, you have 2 copies. And in between the 2 copies, these homologous recombination can happen. And unfortunate homologous recombination can lead to this type of phenotype. So that's another way that mitotic recombination can lead to heterozygosity. So again, cell cycle control, just remember that P16 inhibitor for cyclin CDK, RB when bound to E2F is inhibitor of E2F, which is a transcription factor. When that happens, both RB, P16, loss of function means that E2F is constantly on. Loss of function means cyclin CDK is constantly on. It cannot check the cell cycle. It's going to go from the late G1 to the S phase without stopping. So that's basically the outcome. So here you have the P53 we implicated. Remember that your APC that we have already talked, that this is a proteasomal protein, and that degrade the different types of proteins, and that is also limited. That is also needed for the cell cycle to properly progress. So remember that if you lost the ability to degrade a protein by these APC complexes, which are the proteasomal complexes, if you cannot degrade the protein, that means that it is constantly on. The cell, it will not allow the cell to stop, but it will just drive the cell cycle throughout because you have that active protein. So loss of function of proteasomal degradation will also be a tumor. It's also would cause neoplasticity. So this is a little bit detail of your P53. So just a couple of different ways that the P53 can control these proteins. So remember that a phosphorylated P53 is stabilized, and this only happens due to the damage of the DNA with the ATM and several different things. So one of the important thing here, I just want to mention this, make sure that you understand that P53 by itself helps in the DNA repair by inducing DNA repair, but itself, it's not a DNA repair protein. So it is not a class 6 tumor suppressor, but it is a class 5 tumor suppressor because it controls the cell cycle control directly. So it's a class 5, but it also helps in the apoptosis directly. It induces the apoptosis, and that's why arguably it can be called as the apoptotic protein as well. Because if you just remember and go back to your previous let me show you here. So the definition wise, what happened, cell cycle control proteins, P53 works for that. On the other hand, apoptotic proteins, it is a direct apoptotic proteins which will induce the death. That's why we can call it class 7 as well. But it is by itself, it's not a DNA repair protein. That's why it is not class 6. So this is a concept. Just remember that. All right. So here we are looking at the DNA binding domains. These are all straightforward. Remember that BRCA1 and 2 helps in the DNA break repair if it doesn't happen. And hereditary cancer, breast cancer is the main thing that happens in here. And these are not needed. So let's take a look at All right, so we can do a couple of questions. And all right, let's take a look at this first one. So the first one is a desmosome question. Associated with actin filaments, this is what you need to do if you don't remember and you hate memorizing, is you can always look at that chart. That gives you a clue for this question as well. Good. 15 seconds more. So definitely, I will going to pose the question. So definitely, and I'm not going to pose the answer. So definitely write down the answers. Or you can check from your, if you are in the class, you can check it from your. All right, so I'm stopping it now. All right. So, yep, contain the transmembrane protein desmoglin and the desmocolin. And this is very, very simple. That's why I said right here, you can just remember that desmoglin's desmocolin adherence. So just to make sure that you have this anchoring junction summary, I specifically took this screenshot to show you guys that this will be very important and handy, and you should not miss this type of questions. Because you just, or the thing is that you can find it out, but you will lose time if you have to flip through things or material in doing so. So that's one of the suggestions. All right. So, oh, sorry. Let's look at that right here. Go. So clicker question 2, which of the following characteristic of a malignant tumors? It's an easy question, I think. I can stop it a little bit early, I guess. 1:33. We have a couple of minutes. All right, 15 more seconds. Oh, for first question? Yeah, sure. I will go back to the first question. Yeah. All right, almost done. Stopping it now. Yep, this is a straightforward. It's metastatic, and there is no confusion with that. So that's the only question. Yeah, that's good. So any questions regarding this one? Then I will go back to the next one. Somebody asked me to. So everybody good? If you're not, you can just you can talk to me. Good. Okay. Yeah. Latson, do you have a question on this one?
Yeah, I was just wondering which one was the right answer.
Oh, yeah, the B. Oh, I did not say that.
I missed it.
Oh, okay. So B is the correct answer.
Okay, that's what I thought. I just wanted to make sure.
Yeah, contain the transmembrane protein, desmoglin, and the desmocolin. Yeah, so B is the correct answer.
Thank you.
Yeah, good, everyone? Okay. All right, so clicker question 3. So, oops, sorry. I'm just missing this one. Yeah, so I started it. Sorry about that. So which of the following statement describes the relationship between tumor suppressor proteins and cancers? And all right, I think 53 responses. I'm stopping it now. Yep, that's very straightforward. And just always remember that what we are talking about, that tumor suppressor gene is always a loss of function. That's the only thing that you look for. That will be perfect. There you go. Which of the following statement correctly describe P53? Good. 15 seconds. All right, stopping it now. So here, so correct answer is D. Activation of P53 lead to the inhibition of cycling CDK complexes. So a lot of people actually miss the question. So remember that it is not a membrane receptor that bind to growth factors such as EGF. That is your receptor tyrosine kinase complex. So remember that the P53 is very unstable in the cell. It's a soluble protein, and it only stabilizes to repair DNA if the DNA is damaged, or when the DNA damage happens. Cell division stops until P53 binds to DNA and repairs that damage. The P53 is by itself. A lot of people answered that question. The cell division, again, let me just go back. So I will just spend just a little time here because I think that this needs to be addressed, because you really do not want to mess up this question or the concept. So remember right here, oh, I'm sorry, I'm looking at the wrong one. All right. So right here, you can see that P53 working in different areas, but the important part is that this is how P53 works. So it's stabilized by the DNA damage, and it induces the synthesis of the CYP, P21 CYP, which binds to the cyclin CDK complex, not only arresting G1, G2, but again, G1 to S, S to G2, everywhere. So that's the overall activity of that it is a transcription factor which transcribes or promote the transcription of P21 and inhibits the cyclin CDK complex. So here it is easy. Remember that the question number, some of the people who answered the question right here as the cell division stops until P53 binds to the DNA and repairs the damage. Remember that this is what I emphasize. Remember that P53 by itself right here is not a DNA repair enzyme. It will, so this may lead to a little bit of a confusion here. It is a transcription factor. So this is a transcription. So basically what is showing in this picture, that this is a gene, this is a binding site on the DNA where P53 binds and leads to transcription. So this is a sign, typical sign in molecular biology, which shows a transcription from a transcription factor. Due to that, it will lead to apoptosis, sustained G1, G2 arrest, or the DNA repair. By itself, it will not. So that's the important part that definitely remember in this question, that activation of the P53 leads to the inhibition of the cyclin CDK complexes. And of course, P53 doesn't bind to pre-replication complex promoting transition from G1S. It is not a positive regulator of the cell cycle because it is a tumor suppressor gene. It is not a proto-oncogene where the gain of function will cause the tumor suppressor function. I think that's pretty much it. Okay, good, everyone. Okay, good question. Let me go back. So the question here asks is that what does the answer to question 4 mean, that by a mutation in the TSP gene will cause a necessary gain of function? Oh, mutation in the TSP gene will cause a necessary loss of function in the protein that it encodes? So Garrett, can you talk to me? Are you here, Garrett? Oh, the thing, oh, necessary loss of function. Yeah, I understand. So remember that what the function of the tumor suppressor gene is. The tumor suppressor gene should constantly work and it controls the cell cycle. So that is the necessary function. Necessary function is that a normal tumor suppressor gene will always inhibit, as it is required, the cell cycle. So the mutation of the TSP gene will cause the necessary loss of function, and loss of function and necessary loss of function is not anything different. It is the same thing. That's probably where you probably got confused. Necessary loss of function or loss of function is the same. Necessary gain of function and is the same thing. So that's basically what it is. So think about that necessary means that the normal functioning, it is absolutely required for that tumor suppressor gene to work properly. That's why it is necessary. If it is not necessary, it will cause the loss of function that will it goes. It's probably the wording made it a little bit confusing, I guess. But necessary loss of function or necessary gain of function is the same thing as loss of function or gain of function. Does that make sense, Garrett? Good? Okay, yeah. And those are some of the things that, as I see that sometimes if there is some wording issues with those, and if I really see that that's an issue, I can always address that after your test. And that's the reason I threw out the questions as necessary. So any other issues, anyone? Good? All right, good luck, guys. So that's pretty much your 26th class. That's done. So again, thanks a lot, folks, for tolerating me for the whole semester. And I really appreciate it. Yep, really appreciate it. It was quite a journey. So thank you once again, all of you. So hopefully, and again, I mean, like, it does not mean that once the class is over, that you can talk to me, communicate with email, stop by my office at any time. Once the students, all the time as students. So that's not a problem at all. So down the road, need anything you need to talk, just feel free, not a problem. And hopefully everything is good. Still, there is something in there. So remember that your quizzes, like your differential, your developmental quiz and the summative quiz, I will drop the lowest twos. So if you have a 0 because of you have missed that, and if it is in your grade book, that will be taken care of. So you may see a little bit change. I have not dropped those two. So for each one of them, I will drop the lowest 2. So that will just give you something. All right. And I'm sure that you guys will do good for the 4th exam. It will not be, I mean, like, it is very reasonable. So definitely do all your practice quizzes. That's a good thing. All right. Thank you so much. Really appreciate it. All right. Good luck. Good luck in the summer. Hopefully we will be back in the campus in the fall. Just keeping my fingers crossed. Okay. See you later, guys. Bye-bye.